Introduction to Physiology. Physiology is a branch of biology that deals with the normal functions of living organisms and their parts. Human physiology. Human physiology studies how living organisms work, particularly of the specific characteristics and mechanisms of the human body. Constancy of conditions allows the body to function well. This state is called homeostasis. Homeostasis is the tendency to resist change in order to maintain a stable and relatively constant internal environment. Physiology of the cell. Cell membrane. Cell membrane is the structure that separates the intracellular contents from the extracellular environment. Part of the function of the cell membrane is to protect the cell. The cell membrane also regulates the movement of substances in and out of the cell. The cell membrane also provides attachment for cytoskeleton. The cell membrane also provides binding sites and receptors for enzymes and other substances. And lastly, it allows cell-to-cell recognition. Part of the cell membrane's function is to limit the in and out movement of substances, and there are many ways on how the substances go in and out of the cell. The first ones are endocytosis and exocytosis. Endocytosis is a type of active transport that moves particles into the cell. There are different variations of endocytosis. First, phagocytosis. Phagocytosis, or cell eating, is the process by which large particles are taken in by a cell. It can be large particles, degenerating tissue, or bacteria. The second one is pinocytosis. Pinocytosis, or cell drinking, is the uptake of smaller molecules. Pinocytosis is the process that takes in molecules, including water, which the cell needs from the extracellular fluid. Lastly, receptor-mediated endocytosis. Receptor-mediated endocytosis is a targeted variation of endocytosis that employs receptor proteins in the plasma membrane that have a specific binding affinity for certain substances. Exocytosis is the opposite process of endocytosis, in that its purpose is to expel material from the cell into the extracellular fluid. Waste material is enveloped in a membrane and fuses with the interior of the plasma membrane. This fusion opens the membranous envelope on the exterior of the cell, and the waste material is expelled into the extracellular space. Another way of movement of substances in and out the cell is simple diffusion. Simple diffusion is the movement of substances from the area of high concentration to area of low concentration. Another way is mediated transport systems. This requires a mediator, be it a carrier or a channel. Mediated transport system include the following. First, facilitated diffusion. Facilitated diffusion is the movement of substances from a higher to lower concentration. Facilitated diffusion does not require energy. Another type is primary active transport. Primary active transport is the movement of substances from a lower to higher concentration. Primary active transport requires energy. And lastly, secondary active transport. In secondary active transport, a transporter protein couples the movement of an ion down its electrochemical gradient to the uphill movement of another molecule or ion against a concentration electrochemical gradient, which means in secondary active transport, the movement of substances is also from the area of low to higher concentration. Another way of movement of substances in and out of the cell is through transport channels. There are several types of transport channels. First is the ligand-gated channel. Ligand-gated channel is activated by the binding of a chemical substance, a ligand, to a protein or receptor. Another type is the voltage-gated channel. In voltage-gated channel, changes in membrane potential alters the shape of a protein channel, thus facilitating the movement of substances. Lastly, mechanosensitive channel. In mechanosensitive channel, physical forces affect the cell membrane, thus forcing the channel to open. Detailed discussion about this topic will be discussed in other video. Next is cytoplasm. Cytoplasm refers to the homogeneous substance or cytocell that fills the cell, and is external to the nuclear membrane and internal to the cellular membrane. Cytosol is viscid, translucent and colloidal in nature. It serves as a site for many essential biochemical processes, and it provides a suitable environment for the organelles to perform their function. Cytoplasmic organelles include, first, the mitochondria. Mitochondria are often hot dog-shaped, but they can alter their shape and become rod-like, filamentous and spherical. Mitochondria are known to be the powerhouse of the cell, because it generates most energy needed by the cell to perform its function. Next is the ribosome. Ribosomes are seen under electron microscope as small electron-dense granules that occur singly or in small clusters called polyribosomes or polysomes. They either lie free in the cytoplasm called free ribosomes or are attached to endoplasmic reticulum called attached ribosomes. Ribosomes are sites for protein synthesis, and it is where amino acids are assembled into polypeptide chains. Endoplasmic reticulum. The endoplasmic reticulum consists of two contiguous regions, with the attached ribosomes called rough endoplasmic reticulum and of without attached ribosomes called smooth endoplasmic reticulum. The endoplasmic reticulum serves as a supporting structure for the cytoplasm. It is also involved in the production of numerous substances that are used or exported by the cell. The rough endoplasmic reticulum is involved in the synthesis of proteins, and the smooth endoplasmic reticulum, on the other hand, is the site for the synthesis of lipids. Golgi complex. The Golgi complex consists of several layers of membrane-bound, smooth-surfaced, and flattened tubes that are stacked on top of each other in a semicircular manner. The Golgi complex processes, concentrates, sorts, and packages the proteins it receives from the RER, then release it into the cytoplasmic matrix. Lysosome. Lysosomes are chemical-containing pouches that are made up of unit membrane, and the chemicals they contain are hydrolytic enzymes. The function of lysosome is to digest excess or worn-out organelles, food particles, and engulfed viruses or bacteria. Peroxisome. Peroxisomes, like lysosomes, are membrane-bound spherical bodies that contain chemicals, but instead of hydrolytic enzymes, they contain oxidases and catalases. Lysosomes and peroxisomes are identical and are difficult to differentiate even under microscope. Peroxisomes have two functions: break down fatty acids to be used for forming membranes and as fuel for respiration, and transfer hydrogen from compounds to oxygen to create hydrogen peroxide and then convert hydrogen peroxide into water. Centrosome. Centrosomes are organelles which serve as the main microtubule organizing centers for animal cells. Centrosomes are made from arrangement of two barrel-shaped clusters of microtubules, called centrioles, and a complex of proteins that help additional microtubules to form. The main purpose of a centrosome is to organize microtubules and provide structure for the cell, as well as work to pull chromatids apart during cell division. Nucleus. Nucleus is a membrane-bound structure that occupies the central region of the cell. The key function of the nucleus is to control cell growth and multiplication. Nucleolus. The nucleolus is a round body located inside the nucleus. The main function of the nucleolus is to rewrite ribosomal RNA and combine it with proteins.